It's under. I placed it under.
Okay. Hi, my name is Maia Kinnebrew. I'm a biology major in the College of Creative Studies, and I work with grad student Sonia Hussein in the Department of Chemical Engineering and Dr. Sami Khan in the Department of Chemistry and Biochemistry, and I'm part of the Beckman Scholars Program. And today I'm going to tell you about a 7-transmembrane protein called proteorhodopsin and about how oligomerization affects its function. So here I have a little picture of just 4 proteins complexing together or forming an assembly, and that's what I mean by an oligomer, is an assembly of proteins. So I want you to think back, and I want you to think of a cell, and I'd like to imagine a cell as having this kind of outer fortress, kind of guarding all the contents inside of the cell that make up the machinery that we need for life. And you can see there's little gates in the fortress where you can imagine like a person would stand, a messenger that delivers messages from outside the cell to inside the cell. So here we have a cell, and it's also guarded by this outer membrane, and in order to get messages inside the cell, they have to interact with these membrane proteins. So today we heard a lot about drug delivery in terms of pills, patches, and shots, but I want to think one step deeper and think, how does those drugs actually get inside the cells to interact with the machinery inside the cells to make you feel less sick? And in order to do so, they have to interact with these membrane proteins in these cells. So why are we interested in membrane proteins? For one example, there's a group of membrane proteins called the G protein-coupled receptors, which are 7 transmembrane proteins found in nearly every organ system in your body. So what I mean by 7 transmembrane is the protein is sitting in the membrane, and it also crosses the membrane 7 times, kind of like a string. So here's a schematic of a G protein-coupled receptor showing a drug interacting with the receptor, which then sends a message through some sort of chemical signaling inside of the cell. Some examples of G protein-coupled receptors are the serotonin, dopamine, melatonin receptors. There's a long list, and it's hard to choose which ones I want to talk about. I also thought I was interested in some diseases caused by mutations in G protein-coupled receptors include hypo and hyperthyroidism, several fertility disorders, also carcinomas, which are the most common cancer affecting humans. So they cause all sorts of diseases, and they're very important. So why don't we know a lot about them? And it's estimated to be that 40 to 50% of pharmaceutical drugs on the market today actually target these proteins. But we know very little about them. And the real reason is that they're very difficult to study. So you can see they're found in this complex phospholipid bilayer environment where all of these individual lipids are different lengths and sizes with different bends. And so it's really hard to mimic the native environment of these membrane proteins in order to study them. And most people, to study them, they actually take them out of the lipid bilayer and have them in a solution of basically detergent molecules. And you can see that the lipid bilayer, when the protein's sitting in lipid bilayer, it's in this kind of planar, flat environment, which is very different from this kind of glob of detergent molecules, nonpolar molecules surrounding the protein. So we really want to know what detergent environment best preserves the native function. Does it preserve the native function of the protein? So this is one reason they're difficult to study. Another is these proteins often form complexes. So this is an example of a protein forming a trimer. And we want to know how important is it to maintain these protein-protein contacts. Is this trimer important for the protein function? And these are just some of the reasons why membrane proteins are difficult to study. And to kind of put it in perspective, this is kind of a graph that I just made up actually. I went on the protein data bank and basically you can type in membrane proteins, and you'll see about 300 proteins come up. And they have about like 85,000 proteins actually solved, so this is kind of a conservative graph. But that is less than 0.004% of published protein structures are actually membrane proteins. And all of these other ones are the proteins inside the cell that are running all that machinery. So we know very little about membrane proteins, really just because they're very difficult to study. So for my project, I'm going to be studying proteorhodopsin as a model to study these protein-coupled receptors. We argue it's a kind of a model to help study these G protein-coupled receptors. Reason being is that it's also a 7-trans membrane protein. They tend to form oligomeric complexes, but proteorhodopsin is really easy to express in E. coli compared to these mammalian proteins. We can actually express a lot of it in E. coli, and it's one of the few membrane proteins where we actually know its structure. So this makes it kind of a model to help answer some of the fundamental questions behind studying these really complex proteins, which are, one, what's the best environment for the proteins, and how does the environment affect the oligomeric state, and also, what about the oligomeric state? How important is it for function? So how do both of these really affect the function of these proteins? So a little bit more about proteorhodopsin. It's actually a light-activated proton pump found in 80% of marine bacteria. So it's not really like the messengers where they just kind of send a message. It's actually pumping a proton from inside the cell to outside the cell. And this flow of protons actually creates energy for the bacteria. It's also been found to be primarily monomer and hexamer kind of in a variety of environments. And this is showing a hexamer complex. You can see there's 6 proteins forming this kind of like radial assembly. So this is kind of some of what we know about proteorhodopsin. And for my results, we compared 3 different detergent environments to see how they affect both the oligomeric state and also the function. So first we found that in these 3 different detergents, we get a very different distribution of oligomeric states. So I don't have any graphs, but the non-ionic DDM detergent seems to stabilize the hexameric complexes. So in solution, we see about 90% hexamer when we're using this detergent, versus this slightly different protein, or slightly different detergent with this zwitterionic head group actually stabilizes about 50% monomer and 50% hexamer. So it'd be good if you want to compare hexamer to monomer, you might want to choose this detergent. And we also found that this 2-chain detergent really stabilizes the monomer protein. So it was interesting that they stabilized different ratios of complexes. We also wanted to know, once we separate those complexes, does the different detergents actually affect the function of the protein? So I'm going to talk about this graph in the next slide also, so kind of hang in there. But we can see on the top is basically the monomer protein in all 3 different detergents. We can see that the monomer in all 3 detergents pretty much is behaving like a monomer, even though I haven't told you what this curve is. And the hexamer in all 3 detergents is pretty much behaving like a hexamer. So we can kind of conclude that the detergent environment has less of an effect on the function of the protein than it does on the initial ratio of oligomeric states. So now I'm going to tell you how oligomeric state affects the function of the protein, and so we'll go into a little bit more detail about how it actually pumps a proton. So remember, this protein basically has a proton that hops through this channel, and in order for the proton to hop through the channel, it basically jumps from one amino acid to another amino acid. So this first amino acid that jumps onto it is just an acidic amino acid that grabs this proton and pumps it through the channel. And we can actually determine through a titration experiment, which I'm not going to go into too much detail about, we can determine how often is this protonated. So in this form that you see here, the protein is active because this first amino acid that grabbed the proton does not have a proton, so it's ready to accept a proton, so it's in the active state. And we can basically look at what percent of the time is this amino acid protonated as a measure of function. So we did this, and this is the same graph from the last slide. We can see that we get monomer pKa value of about 8, and this is suggesting that the monomer form is about 50% active. So that means about 50% of the time there's a proton attached to that amino acid, and 50% of the time it's active, ready to pump a proton. And we found that the hexamer is about 90% inactive form. And so this basically, to conclude this, it was interesting because it showed that the oligomeric state really affected the function of the protein. And we can also see that the detergent environment had less of an effect than the oligomeric state on the function of the protein. That's pretty much it. So to conclude, some detergent environments stabilize monomers better than oligomers, and also oligomeric state has a bigger effect on function than the detergent environment does. So the current work that I'm doing, working to reconstitute the protein back into a lipid bilayer, to basically see how how good is how similar is the function when we have it in these micelles to when we have it in a liquid bilayer? Can we compare the function in these micelles to what we would see in vivo? And also we're using a technique called electron paramagnetic resonance to determine details of protein dynamics. It's actually a lot of my work that I just didn't have time to get into today. And with that, I'd like to thank my lab. This is Pond Lab. I want to thank my mentor, Sunny Hussein, my past mentor, Catherine Stone, who showed me the ropes for Sonia, and then my PI, Song Yihan. I'd also like to thank Eric Lewin and the Beckman Scholars Program for making everything possible.